Welcome to G Fun Facts Online. In the ongoing fight against cancer, scientists are constantly searching for a magic bullet, a weapon that can destroy cancerous tumors without harming the rest of the body. Today, we're talking about a candidate for that title, a substance so rare and so powerful, it sounds like something out of science fiction. It's an element called astatine, specifically an isotope known as Astatine-211. This element is the rarest naturally occurring element on Earth, with less than 30 grams existing across the entire planet's crust at any one time. Yet, this fleeting, enigmatic substance is being harnessed to deliver a knockout punch to cancer cells, offering new hope where other treatments have failed. This is the story of Astatine-211, from a prediction on the periodic table to a cutting-edge weapon in modern medicine. So what exactly is astatine? With the atomic number 85, it sits in the halogen group on the periodic table, right below the much more familiar element, iodine. Its name comes from the Greek word astatos, which means unstable, and that name is no exaggeration. Astatine has no stable isotopes, meaning every form of it is radioactive and in a constant state of decay. Its incredible rarity is because it only appears for fleeting moments as a byproduct in the natural radioactive decay of uranium and thorium. The idea of an element in astatine's position wasn't new. Dmitry Mendeleev, the father of the periodic table, predicted its existence back in 1869, calling it iodine. But it took more than 70 years for science to catch up with his prediction. After many false starts and unproven claims, astatine was finally created and identified in 1940 at the University of California, Berkeley. A team of researchers, including Dale Corson, Kenneth McKenzie, and Emilio Segre, used a particle accelerator called a cyclotron to bombard a target made of bismuth with a beam of high-energy alpha particles. This nuclear reaction successfully forged the isotope we're interested in today, astatine-211. 3 years later, scientists Berta Karlik and Trod Bernaire finally found traces of it occurring naturally. Because it's so unstable and disappears so quickly, studying astatine is incredibly difficult. Most of what we know about its properties has been pieced together through clever experiments on highly diluted solutions, and by inferring its behavior based on its neighbors in the periodic table. We know it's a highly radioactive nonmetal that interestingly shows some metallic characteristics. All 32 of its known isotopes are radioactive, with lifespans ranging from a few hours to mere nanoseconds. The longest lived is astatine-210, with a half-life of 8.1 hours. But it's astatine-211, with its slightly shorter half-life of 7.2 hours, that has truly captured the attention of the medical world. Why this specific isotope? What makes astatine-211 so special for fighting cancer? The answer lies in a powerful and precise form of treatment called targeted alpha therapy, or TAT. And for this therapy, astatine-211 is often called the Goldilocks isotope because its properties are just right. The therapeutic magic of astatine-211 comes from how it decays. It releases high-energy alpha particles. An alpha particle consists of 2 protons and 2 neutrons, making it relatively large and heavy on the atomic scale. It carries a significant punch, around 5.87 million electron volts of energy. But the key is how it delivers that energy. Alpha particles have what's called a high linear energy transfer, or LET. This means they dump all their destructive energy over a very, very short distance. Think of it like the difference between a bullet and a cannonball. A single alpha particle from astatine-211 has enough focused power to cause multiple, irreparable, double-strand breaks in a cancer cell's DNA. This level of damage is a death sentence for the cell. To put it in perspective, it's estimated that 1 alpha particle can inflict as much damage to a tumor cell as 10,000 beta particles, which are used in other forms of radiation therapy. But brute force isn't the only advantage. The other just right property is the incredibly short path length of these alpha particles, only about 70 micrometers. That's roughly the width of 2 or 3 human cells. This is a game changer. When Astatine-211 is delivered directly to a tumor, its destructive force is confined to the immediate neighborhood. It obliterates the cancer cells while sparing the healthy organs and tissues right next door. This precision minimizes the collateral damage that is often a major drawback of traditional external beam radiation, which can affect large areas of healthy tissue. Furthermore, Astatine-211's decay is clean and predictable. Each decay releases one and only one alpha particle. This means there isn't a long chain of other radioactive byproducts that could break away and cause unpredictable damage elsewhere in the body. Finally, there's that 7.2-hour half-life. It's the perfect middle ground. It's long enough to allow for the isotope to be produced in a lab, processed, attached to a targeting molecule, and transported to a hospital for administration to a patient. But it's also short enough that the radioactivity fades away quickly, drastically reducing the patient's overall radiation exposure and long-term toxicity. Given that it barely exists in nature, all the Astatine-211 used in medicine must be man-made. The production process is a marvel of modern physics, but it's not without its challenges. The primary method is the same one used by its discoverers, bombarding a target of natural bismuth-209 with a beam of alpha particles inside a cyclotron. This sophisticated process requires extreme precision. One of the biggest technical hurdles is that bismuth has a very low melting point. The intense energy from the alpha particle beam can easily overheat and melt the target, which would stop production in its tracks. To solve this, scientists are designing advanced targets, for example, by making the bismuth layer very thin and bonding it to an aluminum frame that can act as a heat sink, dissipating the heat more effectively. Another critical factor is the energy of the alpha particle beam. The energy must be finely tuned to maximize the creation of Astatine-211 and, just as importantly, minimize the creation of an unwanted sibling isotope, Astatine-210. Astatine-210 is a problem because it has a longer half-life and decays into polonium-210, a highly toxic alpha emitter with a half-life of 138 days. The presence of polonium-210 would introduce significant long-term health risks for the patient, so avoiding its creation is paramount. Once the bombardment is complete, the newly formed Astatine-211 atoms are mixed in with the leftover bismuth. They must be quickly and efficiently separated and purified. Researchers at institutions like Texas A&M University are perfecting methods like extraction chromatography to rapidly recover the pure Astatine-211, ensuring a high-quality final product. However, the biggest bottleneck for widespread use is availability. Production is limited to a small number of cyclotrons worldwide, estimated to be around 30, that can generate the medium-energy alpha beams required. The short half-life adds a ticking clock to the logistics. The isotope must be used very soon after it's made, often requiring overnight shipping to nearby clinical facilities. Despite these hurdles, the actual cost of producing a dose of Astatine-211 is considered relatively modest, on par with other common medical isotopes. Okay, so we've made this potent, cancer-killing isotope. But how do we get it to the tumor and nowhere else? Producing Astatine-211 is only half the battle. The other half is delivery. This is where the magic bullet concept truly comes to life. Scientists attach the Astatine-211 atoms to a larger targeting molecule, creating a drug known as a radiopharmaceutical. This targeting molecule is a biological smart bomb engineered to seek out and bind only to specific proteins, called antigens or receptors, that are found in large numbers on the surface of cancer cells but not on healthy cells. The process of attaching astatine to these molecules is called radiolabeling, and it's a complex chemical puzzle. Because astatine is so unstable, its chemistry isn't fully understood using traditional methods. It shares some properties with iodine, but also acts like a metal in some ways. A major challenge is preventing the astatine atom from breaking off the targeting molecule while it's traveling through the body. This is a phenomenon called deastatination. If the radioactive atom detaches, it won't reach the tumor, and worse, it could accumulate in healthy tissues like the thyroid or stomach, causing unintended damage. To combat this, chemists are engineering new, more robust chemical bonds that can securely cage the astatine atom. For instance, researchers at Chiba University in Japan developed a novel chemical structure that can hold astatine very stably, demonstrating in early studies that the resulting drug accumulates highly in tumors while showing very low uptake in other organs. This brings us to the most exciting part: Astatine-211 in action. The potential of this isotope is being explored in a growing number of clinical trials for a wide range of cancers. One of the most advanced applications is in treating blood cancers like leukemia and multiple myeloma. At the renowned Fred Hutchinson Cancer Center and the University of Washington, clinical trials are using an Astatine-211 radiopharmaceutical called BC8B10. This is an antibody designed to target a protein called CD45, which is found on all blood-forming cells, including cancerous ones. In these trials, patients receive the Astatine-211 drug as part of their preparation for a bone marrow transplant. The targeted alpha radiation sweeps through the bone marrow, precisely eliminating any remaining cancer cells, which creates a clean slate for the new, healthy transplanted cells to grow. Early results from trials with dozens of patients have been very promising, showing that the therapy can deliver a powerful blow to the cancer, while its short half-life minimizes side effects. Another natural target is thyroid cancer. Because astatine is a halogen, just like iodine, the thyroid gland naturally takes it up using the same cellular machinery. This makes Astatine-211 a potential treatment for thyroid cancers, especially for patients whose tumors have become resistant to the standard treatment with radioactive iodine-131. Prostate cancer is also in the crosshairs. Many aggressive prostate cancers have an abundance of a protein on their surface, called PSMA. Scientists are developing molecules that bind tightly to PSMA and are arming them with Astatine-211. This allows for the targeted delivery of alpha radiation directly to prostate cancer cells that may have spread throughout the body. While another alpha emitter, actinium-225, is also used for this, its extremely limited supply has driven intense research into Astatine-211 as a more accessible alternative. The applications don't stop there. Astatine-211 is being investigated for ovarian cancer, neuroendocrine tumors, and in Japan, a Phase 1 clinical trial has even begun for treating rare tumors of the adrenal glands. The journey of Astatine-211, from a mere prediction on a chart to a cancer-fighting powerhouse, is a remarkable story of scientific progress. Of course, challenges remain. The key hurdles are scaling up production and ensuring a reliable supply chain, but dedicated efforts are underway. Networks of cyclotrons, like the Department of Energy's Isotope network in the US, are being established to increase availability. And as chemists continue to unravel the fundamental properties of astatine, they will develop even more stable ways to attach it to targeting molecules. An exciting future direction is the concept of theranostics. This combines therapy and diagnostics. A doctor could first administer a diagnostic version of the drug using a non-destructive isotope, like iodine, to perform a scan and confirm that the targeting molecule is indeed accumulating in the tumor. Once confirmed, they can then administer the therapeutic dose with Astatine-211, ensuring a highly personalized and effective treatment. In conclusion, Astatine-211 represents a monumental leap forward in nuclear medicine. Its unique Goldilocks properties as a powerful, precise alpha emitter make it one of the most promising new weapons against cancer. As research advances and clinical trials provide more data, this incredibly rare and unstable element may soon become a vital tool in oncology, offering profound new hope to patients battling some of the world's most difficult diseases. The once theoretical Ika iodine is poised to make a very real and very powerful impact on human health. Thanks for tuning in to G Fun Facts Online.